Hello, Peter. There seems to be some connection issue, maybe. Peter, can we please ask you to hang up and then dial in again? And we apologize with everyone. We should just take a couple of seconds. Hi,
Hello?
Peter, we can hear you now.
Ah, very good. Okay. I apologize for that technical.
No problem at all.
It had been working fine before. Anyway, I just wanted to just begin by thanking you very much for that thorough introduction, both to Magda and Alessandra, and also wanted to welcome all the webinar participants and thanking you in advance for your time and interest in this really important call topic, addressing a disease with huge unmet need. And a lot of accompanying morbidity and mortality. My co-leader is Dr. Aimo Kant from Sanofi, and together Sanofi and AbbVie are co-leading this project. And the intention for this webinar is, as Magda had outlined, to review several aspects of this proposal, including the rationale, including the deliverables, possible work package structure, what would be expected of applicant consortia, and the impact of the overall project. And we look forward to vigorous discussion with all of you, and starting with the question and answer session at the end of this presentation. So just to get started, then with this slide that you can hopefully see on your screen, the need for public-private collaboration. I mean, this, in my mind, is these next two slides really go to why should we undertake this kind of a project in diabetic kidney disease, and why is it that a single entity, like for example, a single academic institution or a single company, why is it that a single entity can't undertake this just by themselves? So let's start with, what does the clinical landscape look like in diabetic kidney disease? I happen to be a transplant nephrologist by training, so I've treated many of these patients. And the fact is, the standard of care really hasn't changed since the beginning of the last decade, when ACEs and ARBs, that is angiotensin converting enzyme inhibitors and angiotensin receptor blockers were introduced in the early 2000s. They, as we all know, who practice in this field and who do research in this field, they have demonstrated efficacy at slowing the disease somewhat, but a lot remains to be done, and that's really highlighted in the second bullet. There's still a very unacceptably fast rate of progression of many of these patients to end-stage renal disease. But in point of fact, most of these patients actually never reach end-stage renal disease because they die usually of congestive heart failure or some other cardiovascular or infectious disease cause before they even reach end-stage renal disease. So, in order to undertake research in this area, when we think about the recently completed and unfortunately failed trials in this field, many of them have failed because of unforeseen safety effects and maybe also a non-appreciation or lack of a full appreciation of efficacy that didn't get borne out in the phase 3 approval trials. And the reason for that, we think, is a lack of highly predictive biomarkers, both from a prognostic standpoint and a predictive standpoint, including dynamic biomarkers, and we'll be getting more into this later in the future slides. There is proteinuria, albuminuria, that is very much used today in the clinic to help guide therapy and manage patients and assess disease severity. but there are significant problems associated with albuminuria, including substantial intra-patient variability, and really there's a plateauing effect, if you will, in terms of its ability to predict hard outcomes, i.e. at lower levels of albuminuria, yeah, there's a decent correlation between albuminuria and hard outcomes. But it's not linear. When you go to higher levels of albuminuria, at a certain point, further increases in albuminuria don't necessarily connote higher risk, and that's very well documented in the literature. So because of these two main problems with albuminuria, we really do need better biomarkers to make our clinical development and clinical trial enterprise, as well as our preclinical model enterprise for diabetic kidney disease, much more efficient than it currently is. Let's go to the next slide, please. So, thank you. As I've already hinted at, it's going to be important to identify and validate new biomarkers for renal function. If you look at the current literature, some ideas about what these biomarkers might be have already begun to emerge. And I won't go into any specific detail, but now we can do that in the question and answer. But suffice it to say, none of these are really validated at this point, and to my knowledge, none of them have really been put into large-scale clinical trials to really help materially with improving patient selection. And by the way, just a quick word with another commonly used phrase, personalized medicine. Identifying the right patient population also helps us to personalize the therapies, because we're better able to predict with biomarkers, number one, the patients who are more rapidly progressing and therefore have a greater need for help with a new therapy, and number two, identifying those patients that are more likely to respond to the new drug therapy or whatever the new therapy is. So in order to make material progress in this field, it's our belief that we really have to collaborate with one another within a network of medical institutions and industry, including large companies like the companies that IMO and I belong to, Sanofi and AbbVie, but also smaller companies as well, small to medium-sized enterprises. And the IMI framework is exactly set up to serve as a platform, a coming together place for just these kinds of collaborations to be formed. And so what we really are looking for are in any applicant consortium, and we'll talk about this more, but also from us, the FPIA companies involved in this project, is expertise in biomarkers, bioinformatics, preclinical in vitro and in vivo diabetic kidney disease models, and finally, expertise within the renal imaging area and making connections with renal biopsies. And we'll get into that in a minute. But suffice it to say, this is, as they say, this is really going to take a village. It really requires collaboration. Next slide, please. So the objectives, the high-level objectives for this full project are really fourfold, and they're here on the screen. I'm not going to read them to you, but I'm going to talk about them. The first one really has to do with longitudinal cohorts and databases that describe the patients in these longitudinal cohorts. Just to say, this disease, diabetic kidney disease, doesn't necessarily progress at a constant rate. So the cohort that you're talking about here has to be of sufficient length over time, of sufficient continuity over time to capture the variability of progression, and so that patients that on average progress faster can be differentiated from patients that progress slower. And ideally, we'd want to do this for type 1 as well as type 2 diabetic patients, understanding that there are obviously many more type 2 diabetic patients than there are type 1. So the cohorts really need to be large enough, hopefully, to answer the question for both sets of patients, and even really to see if there is a difference. It may be that there is no real difference between how type 1 and type 2 diabetic patients progress. It's never really been exactly answered in the literature. When we look at these longitudinal cohorts and we extract from them biomarker candidates, and what we want to do then is replicate these findings in other cohorts and really to validate them. When we talk about developing new therapies in diabetic kidney disease, this will inevitably involve the undertaking of global trials. So whatever the biomarkers are that we identify, we want to make sure that they hold up to that they're able to select out the proper patients, whether we're talking about a Chinese population, a population in Germany, or a population in the United States. And hopefully with the same biomarkers at the same weighting. It's also I'll just add this, as another sort of insight that we share as a group, is that it's more than likely that it'll take more than one biomarker. We would envision that the likelihood is that we'll end up needing to form a panel of biomarkers in a statistical model to have the best chance of pulling out these patients successfully. So whatever we learn from the clinical cohort, what we want to do is back translate these clinical findings to develop and prioritize, in vivo and in vitro models of DKD. As many of you know who work in this field, there's really no one animal model of DKD that directly pegs to the clinical condition. There are models, that do describe and seem to correlate with some aspects of DKD, but there's no all-in-one model. And so the question is, are the pathways that are operating in the animal models, are they the same or are they different from the ones that are operating in human clinical disease? To the extent that they're the same, we want to lift those animal models up higher on the priority ranking, and to the extent that they're very different, we probably would want to deprioritize them. But we don't have the biomarkers right now to do that as effectively as we should, and so that's one of the key goals of this project. And then finally, we want to take advantage of the fruit of all this labor. When we have the novel biomarkers validated and where we have animal models that describe the disease, we'd like to plug that into clinical and preclinical development in order to enhance the development process from its current state. Next slide, please. So, hopefully, even just from my introduction, hopefully it's apparent to everyone on the webinar that we're really talking about a pre-competitive type of project. This is the kind of, hopefully you can see just from my introduction, that the findings of this kind of project would be helpful to folks that are in an academic laboratory, companies that are small, companies that are large, that are undertaking, development and or basic research in this disease. And so from that standpoint, it shouldn't really help one competitor, if you want to call them, or one institution, more than the other. It should, as the phrase goes, it should lift all boats. And again, what we're after is, what's in these 4 bullets: identifying patients at high risk for progression, what we would call fast progressors, identifying subpopulations that are likely to respond to a candidate therapeutic, monitoring the response to treatment with dynamic biomarkers and being able to predict that in earlier stages of development. I'll just briefly add to this point, and really this goes to the first 3 as well. When industry at least undertakes a phase 2 and phase 3 program, we would really need to rely, I would say, heavily on well-validated biomarkers to help decide whether or not the drug is likely to be efficacious and safe, and whether or not to undertake a phase 3 program. But I think that's just one of the benefits, of undertaking these, the search for these biomarkers. I think we get an enhanced disease understanding as well, and an increased appreciation for which animal models really most closely mimic human disease. And then this last point, boy, this is the issue that has really sunk a lot of trials, including the ASCEND trial from Avastin, the Beacon trial for bardoxolone, the VA Nephron study, the SUN trial for solidexide. Boy, I could name several trials, that have really been sunk by unforeseen safety issues. And gee, what if we had biomarkers in earlier development that we could use to help sort out new molecular entities for their potential to induce safety issues or even help. So for example, if you had a biomarker that helped us to predict the risk of congestive heart failure, it's not just a matter of getting bad news out of that biomarker. It might be that this new therapy helps with congestive heart failure, and a really good biomarker might be able to help you pull out that benefit as well. And just a quick word about regulators, global regulators, whether you're talking about the FDA, the PMDA, the EMA, the Chinese FDA, they're all very interested in cardiorenal safety issues. Whether there are safety issues anywhere in that spectrum, whether they be congestive heart failure issues, myocardial infarction, stroke, etc. All the past trials have really borne out the need to look hard at safety. And so, again, as the final summation statement says, if we make progress on the above four goals, we'll really have made a huge impact to this field, which really needs it because these patients have a huge mortality rate, comparable to very, very serious cancers. Next slide. So I'll try to move a little bit faster now. I wanted to set the stage, with a little bit more words, on these earlier slides, so that it hopefully makes sense to everyone. Hopefully, a lot of the information on this slide I've reviewed. If we're successful in this project, we will understand the pathophysiology of diabetic kidney disease a lot better than what we do. We will improve the translatability from preclinical disease to human disease, and help us make assessments of early drug candidates, even in the animal model stage, for whether they should be taken into the clinic. And then, from a clinical standpoint, we should be able to become a lot better than we currently are at improving stratification of patients regarding the state of their disease, the responsiveness to and benefit from novel drug approaches. And then, going along with that, because we're able to identify patients that are progressing more quickly, that means that the placebo event rate is going to be a lot higher. And placebo event rate, as many of you know, is a critical driver in overall trial size and cost. So that's important for us in industry when we think about pitching these new trials that we come up with to our senior management. One of the things that does get looked at pretty hard, as people in industry who are on the call know, is the size and more importantly, the cost of the trial. And then, the other issue is, health technology assessments that get done globally by various payers all over the world. They want to know that we're really impacting important things. Not just movement on a biomarker, but movement on some kind of really important hard outcome. So traditionally, that's been need for dialysis, or the ability to delay dialysis, end-stage renal disease, the ability to stave off the need for a transplant, which globally are in short supply. So if we had biomarkers that could help health technology assessments and help these payers make these assessments more precisely, that also is a huge benefit to the field. Next slide, please. This slide really talks more about, we're starting now to get into the high-level structural requirements, for the collaboration. And, what I'm going to show you in a couple of slides is a first assessment or a first suggestion at what the work package is of this project might be that would satisfy the objectives I've already reviewed. The idea is that every team that's working on work packages would consist ideally of academic and industry members. And there would be frequent interactions to make sure that there's good knowledge exchange. We would have frequent, with a sensible frequency, board meeting frequency to make sure that we have good knowledge transfer between the teams and that we're able to work well together. A big part of the future plan of this project would be that we could take the fruit of whatever we found out and take that to regulators. Regulators, I would say, maybe I can't speak for all therapeutic areas, but I would say, globally, both the EMA and the FDA have demonstrated a real willingness to be receptive to new data sets and even to adjust their approval, adjust their approval criteria based on those new data sets. The problem is we don't have an awful lot of new compelling data saying which biomarkers should serve as potential surrogate outcomes in clinical trials. And then we'd want to make sure we have a structure in place for disseminating the knowledge gained in this project, but also, as you'll see, a couple of the work packages really have a longer term aspect to them, and we should think about the sustainability of this project because once we set up the and take advantage of the infrastructure within this project, I think this is something that for several years and maybe even decades to come could continue to bear fruit. And so we need to think about the longer term of this project, as I'm sure will become apparent to you as we march through the deliverables. Next slide, please. So this is a suggestion for how the work packages might be divvied up. I'm going to jump around a little bit, start with work package number 6, just to get that out of the way. There's several administrative tasks that IMO and I will need to deal with as we in conjunction with Magda and Peter Hecht and others. But just to say that will be put into a separate work package. But the technical meat of this project is really contained in the first 5 work packages. Number 1 is to go after this whole issue of fast versus slow progressors, for diabetic CKD. When we think about fast versus slow progressors, we're really thinking about how fast patients progress through stages, typically stages 2, 3, and 4. There's also interest in earlier stages, obviously, and we're going to get to that with work package 5. But for this part of the project, really what we'd want to see is to look at currently available prospective cohorts, and maybe if it makes sense to develop our own prospective cohort as well. But first to look at currently available prospective cohorts. And these cohorts, we have a couple of requirements, things that we're looking for. We're looking for them to be large enough, at least 500 patients. Our FPA, that's the consensus of the FPA members who are involved in this project. And at least that these patients be followed for a minimum of 3 years, really preferably 4 or 5 years. Again, this is to capture the variability of progression and to be able to draw a straight line over sometimes a curvy path of progression. Really when we think about progression on a sort of on a more micro scale, we think about how fast the estimated GFR is decreasing, a GFR loss rate. And so really with these cohorts, the first thing that we're going to try to assess is how fast are different patients moving through these CKD stages, and then, to have access to these biobank samples so that we can study an array of other potential candidate biomarkers that we already, several of us already have in mind, but we're always looking for new suggestions from the selected, applicant consortium. What candidate biomarkers should we evaluate in these patients to help us differentiate the fast from slow progressors? As I stated earlier, this is a critical this is a critical facet to this project. It's very important that we understand what differentiates fast from slow progression. Work package number 2, now we switch gears a little bit and we think about what are predictive biomarkers of efficacy response. So there are obviously, as I already mentioned, there are ACE inhibitors and angiotensin receptor blockers that have already been approved. That might be a first trial site, if you will, first workhorse, if you will, to try to assess for new predictive biomarkers and whether or not they can work. It may well be that some of these predictive biomarkers could carry over into novel biomarkers. Into novel mechanisms of action. And so we call these, some of these predictive biomarkers would be things that we would follow over time, i.e. they're dynamic. So they change in their values as patients progress, and they predict as well, the likelihood of future fast progression. And once these biomarkers are identified, hopefully that will point to a specific pathway that we can then turn our attention back to preclinical models, let's say back to rat and mouse models of diabetic kidney disease, and ask, are these pathways operating and are they key drivers of disease in preclinical models? And so that's, again, that's sort of the next idea there, identifying pathways and mechanisms. These are all closely related sub-goals within the second work package. We tacked on their safety biomarkers as well. Some of the dynamic biomarkers may be biomarkers that indicate risk for future safety, or again, as I mentioned, improvements in safety with regard to specific events like congestive heart failure. That would be fantastic if we could identify some of those safety biomarkers. Work package number 3. We'd want to then, after we've identified these candidate biomarkers that have been done in work package 1 and 2, is to take whatever has been identified and really validate them, in other prospective cohorts. Ideally, and we could also do them within the setting of registration trials. We could use this information to develop companion diagnostics, and again, that would involve conversations with regulators, because we need, obviously, their approval of whatever companion diagnostic we come up with. Typically, the companion diagnostics wouldn't be the prognostic biomarkers; they would be that predict rates of progression. They would typically be the predictive biomarkers. And then, obviously, we'd want to get the word out not only to regulators but to payers, and that's all part of work package 3. And work package 4 really talks about the connection between imaging and biopsies. And this was suggested by one of the members originally in our group. Is this is a fascinating connection that really hasn't been explored as well as it could be. We fully realize that not everyone by any stretch gets biopsied who has diabetic kidney disease. In fact, most patients don't. However, if there were, in the few that do, if we had access to just a few of those biopsies and we're able to match those samples with images that were taken of the patient's kidneys at the same time, there might be biomarkers that we could get out of the images and correlate them with features on the biopsy. Again, this could help in a number of different ways. It might suggest prognostic and predictive biomarkers, but also could really help deepen our understanding of DKD pathophysiology. And then work package 5, is really talking about now thinking earlier on than work package 1, really thinking about, which patients amongst the universe of patients that have diabetes, be it type 1 or type 2, about roughly a third of those patients, just a third, although it's a pretty serious thing if you're one of those patients that develops kidney disease. But roughly something like a third of those patients will go on to develop diabetic nephropathy. And all the advances, the other key thing to remember about this is that, with all the advances in diabetes therapy and cardiovascular care, many of the diabetic complications have seen dramatic reductions in incidence in terms of cardiovascular disease, even neuropathy, but the one and diabetic retinopathy, many of those have decreased. The one disease complication of diabetes that has not decreased despite all these new therapies is diabetic kidney disease. So there's obviously some important mechanism that hasn't been uncovered yet, a key determinant of if you're a diabetic, will you develop diabetic kidney disease, and will the diabetic kidney disease process get driven in a particular patient? So work package 5 is really aimed at identifying what those markers would be, and in seeing if there's any differentiation between type 1 and type 2 diabetes. Next slide, please. Now we get to you, the prospective applicants. So when we came up with this slide, IMO and I were considering, very much the capabilities of the FPIA companies that are already involved, and sort of what the key missing pieces would be that applicants could come in and fill in very, very nicely. And that's what's really listed here. And you'll see that there's a pretty good match between, what we'd like to see from a contribution standpoint and the objectives set out earlier in the earlier slides, as well as what the work packages actually are. So you can see pretty clearly, for example, that the first bullet here of what we're looking for is to go after, or it would be to favor applicants that have real access to stored serum, plasma, and urine samples from the type of clinical cohorts that we're talking about. Again, ideally over 500 patients and at least 4 years of follow-up, and that these samples be linked longitudinally to clinical phenotypes, including the serum creatinine. Cystatin C is very, very important, as many of you know, as a key marker of kidney disease, proteinuria, and end-stage renal disease, and that it's obtained, that the data set is well put together, and the samples are collected at regular intervals and have been well maintained. The second one, the second bullet really relates to access to diabetic kidney disease renal biopsies. Again, access to these biopsy samples that have been well maintained, ideally that were obtained at some degree of intervals, some degree of regular interval, longitudinally over time for probably what would likely be a smaller group of patients, but that's okay. I don't think we need nearly as many diabetic renal biopsy samples as we would, serum and plasma samples. But at least well enough preserved to enable RNA protein epigenetic extractions and analysis and/or correlations with imaging analysis, which means ideally, the samples that we would have access to through the applicant would be accompanied by concomitant and concurrent imaging studies, whereby candidate imaging biomarkers could be evaluated for their ability to correlate with biopsy features. And then the third bullet really goes to expertise with respect to animal and/or in vitro models. I mean, this goes to this whole issue that we talked about earlier with respect to there is no ideal animal model, and but some animal models are going to be better than others, and we already have a sense of that in the literature. but can we use these new biomarkers to prioritize which animal models correlate to which aspects of disease, and which animal models are should be the go-to animal models for testing of new therapeutics. And then, we need also, imaging and serum plasma, urine biomarker expertise, large-scale data management, and computational expertise. The overall hope and really the expectation is that once the applicant consortium is selected, that we all work together really, really well, that there's real synergy. I know that's sort of an overused word, but if we're not working together well, that could get in the way of having a really productive project. So the idea would be that, part of our thinking, ideally, in selecting applicants would be partly an assessment of, can we work together well and be productive together. Next slide. So you can see here listed on this slide who are members of the FPA consortium that have already made commitments to this project. Aside from IMOs and my company, there's Eli Lilly, Novo Nordisk, Bayer, Merck, and the Juvenile Diabetes Research Foundation. So I hope you can gain an appreciation that these are not small players. These are major players in the field of drug development, and they're global enterprises. So I think this diabetic kidney disease call topic has been taken already very, very seriously by members of these companies and also by their managements. And there's a strong desire to move forward. So hopefully that speaks to that there is the idea that there is a commitment. And that all goes back to what I mentioned at the start of this call, which is that there's a huge unmet need. These patients have, having taken care of these patients, I can tell you from personal experience, they have pretty dire comorbidities that interfere with the quality and the quantity of their life. Many of them will end up needing dialysis because there aren't enough transplant patients to go around. And on dialysis, the length of your life is greatly curtailed. The first-year average mortality rates in the United States and really elsewhere is on the order of 20 to 25%. So that's high. As you may know, that's comparable to many of the most deadliest metastatic cancers out there. So hopefully the gravity of this patient population is impressed, upon you, and hopefully this project will make sense to you and garner your interest and sustain your interest, and we look forward to that. So, we've been able to, what was submitted to the IMIJU was an in-kind contribution of over 14 million euros. Again, my goal here is to not talk about administrative, matters, but I think it's public knowledge that the IMI will typically match that in an in-kind cash contribution. So, I think this is an ambitious project. I think that this is a good amount of capital and in-kind contributions that will enable us to make a significant dent in the unmet need of this disease, the unmet knowledge gaps that exist that prevent us right now from conducting clinical trials as efficiently as we could. And so that's why we're very excited to bring this forward to you for your consideration. And there's one more slide, is that right? Or was that the last one? Oh, there's a couple more, I guess. What's in it for you? So of course we have to think about you and what your interests are. Hopefully this slide, and we're happy to talk about this more in the question and answer session. But obviously, the IMI is set up, in my opinion, in a very smart way, whereby industry makes in-kind contributions, and the big companies are not the recipient of cash, but the academic researchers that sign on to specific projects, and the small and medium size enterprises are direct beneficiaries of funding. And there's the chance, if you consider this a benefit, to collaborate with industry partners. And I think it's common to most people's experience, and we can talk about this further, that when people are working together well, there's really a work multiplier effect. That really, at the end of the day, 1 + 1 is really greater than 2 when it comes to brain power, bringing brain power to bear on some of these critical issues. I would say an additional benefit for at least the small to medium-sized enterprises, maybe for academic researchers too, is a seat at the table when we go to bring this information to global regulators and payers during negotiations regarding these newly identified and validated biomarkers. So I think I think this work multiplier effect is very important. I think you'll find that if you apply and are selected, you'll be part of a very diverse group of stakeholders, each with different experience. And at least in my experience, when you bring together people that have very different but still relevant experiences, that's a huge benefit for any project. And I think IMI, and in particular this DKD call topic is set up to deliver exactly that. Next slide. So yeah, so now that we've seen the work packages, and I'm going to run through these a little bit faster. The work packages are really set up to deliver things. And that's, at the end of the day, what we're all really interested in is delivering new knowledge that is material and is beneficial to the research enterprise in diabetic kidney disease. And you'll see very familiar words on this slide. I won't read them to you, but hopefully you can appreciate that there's a very strong link between the suggested work packages and these deliverables. And you see, common words that we've used before, like the dynamic biomarkers, correlations between imaging and renal biopsies, identifying new and validating new prognostic biomarkers. And the idea is this will be applied to not only early development but also to registration trials. Next slide, please. And then, we haven't forgotten about safety. We've talked a lot about this in this, in this webinar, especially cardiovascular, biomarkers that would help us predict early on what's the impact of this drug, good or bad, on cardiovascular outcomes. Boy, if we had that information early on, that would really make our development process a lot more efficient. And then, another key deliverable is, and this really, the second bullet here, something I've talked about, developing these and taking advantage of leveraging prospective cohorts that are big enough and have been followed, where patients have been followed long enough to have them be beneficial. This is really something that, this is one of the goals, I think, that we can talk about from a sustainability standpoint. As the project progresses, we can think about how could we maintain and grow these cohorts over time so that they can continue to bear meaningful fruit for this field, over several years, maybe going on into decades. I think that would be a really good thing for the group to think about once we're all formed. And then, I already talked about this. Once we have this great, new knowledge with respect to identified and validated biomarkers, is communicating this to other very much interested stakeholders, the regulators, payers, but also to the physicians, the medical care workers who care for the patients, and really ultimately the patients themselves. Next slide, please. And then, I won't read this, but hopefully what's here on this slide is evidence that we want to take what we learn in the clinical arena and out of the clinical cohorts and work that back to helping us to pick out and prioritize these preclinical models, figure out what are the corresponding driving mechanisms, which is really lacking in this field. And so that way we can lift the whole field by undertaking a project like this, not only the clinical piece but also the preclinical piece, and then differentiating, if there is a differentiation to be made between type 1 and type 2 diabetic patients. Next slide, please. So that's everything that we have prepared, and I probably should have waited a little longer at the end of each slide to let Imo talk, but maybe I'll stop right now and just ask if Imo has additional comments that he wanted to make as the co-lead on this project, and we can go back to any slide that Imo would like to talk about, and then we can just, if it's okay with Magda and Alessandra, open it up to general questions.
Thank you very much, Peter, for your presentation. Now, as you mentioned, it's time for questions and answers. I would just like to remind everyone how to ask questions. You can do so in writing using the question panel. Just type your text in the panel and click send. Or if you want to speak directly with Magda or with Imo or Peter, just use the raise your hand button, the little yellow hand button, and we will unmute you. So we already have one question by Lina.
Oh, I'm sorry. I just sorry. Before the question, I wanted to see if Imo had any additional comments. Imo, did you have anything you wanted to add? Imo still on? I don't know.
Imo, can you hear us? I see you are unmuted from here. So, should there be any issue with your line? Also, the best probably. Again, as Peter did before, to hang up and then dial in again. I'm sorry about that. It's just usually the best troubleshooting system we have for webinars, the best tip. So I know if you're trying to get in touch with us, but we can't hear you, please hang up and dial in again. Or you may also just be muted.
But while he's doing that, if we want to go to questions, to start questions, we can certainly.
Yeah, maybe we can go on with the first question, and while we wait for people to send in more questions, Peter. Then we will see if Imo is with us again. I see that he's redialing right now, so he's going to be with us in a few seconds, I guess. So Lina Christopoulou, was asking, how can you get biomarkers out of images?
Right. So my first statement will be, I'm not a radiologist, so I'm not necessarily the imaging expert, but I do know that there are features. We've had several of our imaging experts tell us this, that there are specific features, let's say in an MRI that you take of the kidney, in T2, T1 weightings, that you can evaluate. Really what we're talking about here is evaluating what was thought by many to be a final common pathway that not only diabetic kidney disease patients enter, but also patients with other causes of other etiologies of CKD, like, for example, FSGS, focal segmental glomerular sclerosis, or IgA nephropathy, or lupus nephritis. All these diseases seem to converge on a common phenotype, if you will, within the kidney that's characterized by fibrosis. And there's glomerular fibrosis, cortical fibrosis, but then there's also going to be medullary fibrosis, and that could correlate. That really does correlate. I think it does, but we would need further data to develop this. Tubular interstitial fibrosis. And tubular interstitial fibrosis, in the kidney has been well documented to correlate with GFR loss. So the question is, and you can pick up fibrosis on images. And so the question is, are there quantitative measures of fibrosis on an imaging scan that you could correlate with either disease progression and/or biopsy features? Did I answer the question?
Lina, let us know in case you have any follow-up questions or any other doubts about this point, and if you want, we can also unmute you, and then you can speak with Peter directly. In the meantime, I see that Imo is back online, so Imo, would you like maybe to add something further on to Peter's presentation?
Yes, so first of all, I would like to thank Peter for this very comprehensive presentation, which was also easy to follow and even kind of entertaining, I would say. I was glad, not to interrupt him after the slides, and I only have two comments. One refers to the project architecture, and this really is only a suggestion. So it is the overall project architecture, the work packages, the tasks assigned to the work packages will, of course, depend on the nature and the composition of the winning consortium at the end. So it is the current proposal is based on the overall objectives of the project that have been outlined by Peter here, but other designs are possible, and we are open here to other proposals. So that was one thing, and I have an addition that I would like to make to the "What's in it for you" slide. So the consortium, or the academic groups that are part of the consortium, will not only gain something from this project in terms of funding or they're seeing the multiplier effect of this collaboration, but of course, and Peter has addressed this sustainability effect, and this does not only relate to the cohorts that will be studied, but also to the network that's been formed. So I think participation in this project may also set the stage for future collaborations and will expand the network of all participants in a way that this can be utilized after the project has been finished. So these were the only two comments I wanted to make, and again, thank Peter for this very nice presentation.
Thank you very much, Aimo. And this was Aimo Kant from Sanofi, part of the FPA consortium that will participate in the project. So while we wait for some questions to come in, maybe Magda, is there anything you would like to add?
Yes, as well, I would like to thank both speakers for the presentation and all the details. I'm sure it was very useful for all the potential applicants. I just wanted to mention that the indicative deadline for applications for this call is 13th of October. And as you can see on our website, the draft, pretty much almost final text, is already on our website, and official launch should happen within coming days. So you can already start working and assembling the consortium and start thinking about your short proposals. Other than that, I really don't have anything else to add. Hopefully you will have some questions, and if you don't, I guess Peter's presentation was very comprehensive, and all the information was provided. In any case, if something comes to mind after the presentation, please don't hesitate to contact us by email, either directly to myself or to the info desk. We will be happy to provide you with further information. Alessandra, do we have any other questions?
No, we don't. We just have Catherine from the audience that says she would really, really like to find the consortium. So Catherine, let me remind you and everyone else, the best ways to network here is you can first of all use the list of participants that we will share with you after the webinar. We should share with you today or tomorrow at the latest. You can simply contact any other fellow webinar participants that you think may be relevant for a consortium formation. Then we have a partner search tool on the IMI website, as well as another tool that we recommend. You will also receive these links in the follow-up email that we send after the webinar. We, of course, as usual, advise everyone to network with your contacts, with all the people you know working in the field. And for any question, as Magda said, never be afraid to contact us by phone. You can quickly pick up the phone and call the IMI program office, or by email, sending questions to infodesk@imi.europa.eu. Another good way maybe to find some partners is to use the IMI LinkedIn group. You will find the link from the homepage of our website, because it's very likely that in the same group are people with the same interests and the same scope for an application as you are. So as I see no other questions coming in, I think it may be just that Aimo and Peter were super clear, and there's no room for doubts by now. The call will be launched very soon, as Magda said. At any stage, contact us for any questions you may have. Please refrain from contacting Peter and Aimo, as well as any other FPA partners or associated partners in this call topic, and contact the IMI program office in any case. So thank you very much, everyone, for your participation. Thank you very much also Peter and Aimo for preparing the presentation and presenting the topic. Thank you, Magda Gon, who will follow the project once the project is launched. Today it's also Magda's birthday, so happy birthday to Magda. And we wish you all the best for a great proposal submission. And do not forget that we're always here for any questions you may have. Thank you very much, and goodbye.
Thank you. Bye-bye.
Bye. Thank you. Bye-bye.